Good morning. Welcome to Tomocube webinar series. And today I'd like to talk about the principle of a holotomography system, and also I'd like to share how it can be used for the study of the platelet. My name is Hyun-Kun Park, the professor in physics KAIST and also co-founder and CTO of Tomocube. Let me begin by expressing my gratitude for your attending here, and also I'd like to start talking about the principle of conventional microscopic imaging technique, which is crucial for the visualizations of cells and tissues, and as well as the assessments of the cell for biological study as well as clinical diagnosis. Conventional microscope technique include bright field microscope, phase contrast, or fluorescent microscope, including 2D and 3D confocal microscope. And even though they can provide very valuable information of cells and tissues, but they have several limitations. First, you have to worry about sample preparation. The typical procedures for microscopic sample preparation usually requires fixation, staining, or the introduce of exogenous labels such as dyes, or sometimes you have to make the cell expressing the fluorescent protein by the transfection procedures. By doing that, the cells are not anymore live, and you have to worry about the side effects of the staining procedures, including phototoxicity or the photodamaging, etc. And even though they can provide very good molecular specificity, the process is very long and very complicated. And the conventional microscope technique, even though they can provide good imaging contrast, but they only provide qualitative information of the cell. So what I mean by that, even though you have high contrast in bright field stained images of tissues, but you do not have information in terms of quantitative numbers, such as cellular volumes or the concentration and dryness of cells or subcellular organelles cannot be retrieved in physical numbers. And since they all use optical illumination, the resolution, the spatial resolution is limited by the diffraction limited spot size, typically 200 nanometers or the 300 nanometers. And this is typo, sorry about that, 200 nanometer. And temporal resolution is also limited by due to the low signal to noise ratio, and sometimes it needs the scanning of the illumination and detection, so it takes a long time to measure two-dimensional field of view. And there are other techniques such as electron microscope or the atomic force microscope. Even though they can provide very high spatial resolution, but those are not very well suitable with live cell imaging because they require metal coatings of the sample, or sometimes it takes very long to acquire two-dimensional field of view. So it is not ideal to measure the dynamics of the live cell. The holotomography system, which is the name of the principle that has been commercialized by Tomocube, enables label-free 3D images of cells and tissues in terms of quantitative parameters. The principle of HCT can be easily understood as the optical analogs to X-ray CT computed tomography system. As you may know, the CT require the measures multiple 2D X-ray images as a function of illumination angles. And from those multiple 2D measurements from different view, then the three-dimensional tomograms of human body can be retrieved by reconstruction, by performing reconstruction process, which is basically the inverse of the X-ray diffraction projection. And by doing that, the clinicians can look at the organelles of human bodies without invasive procedures. HCT is optical version of X-ray CT. It measures multiple 2D hologram images of live cells, and then by scanning the illumination angles within some of the ranges, then you can measure multiple 2D holograms as a function of the illumination angles of the cell. And by applying the three-dimensional reconstruction algorithm, you can retrieve three-dimensional tomograms of live cell. And then the difference between X-ray CT and HCT is that the X-ray CT provides the X-ray absorptivity as the imaging contrast, whereas HCT provides refractive index distribution in terms of intrinsic imaging contrast. Refractive index determines the speed of light in a medium. So if you have higher refractive index, the speed of light will be decreased, and that is the intrinsic optical contrast in HCT. So the realizations of X-ray CT and HCT has been demonstrated by the different methods by different groups, but the conventional X-ray CT utilize rotations of source and detectors using mechanical motors, and they can realize three-dimensional scanning. But in this case of the Tomocube HCT machine, it used the DMD or digital micrometer device in order to systematically control the illumination angles without moving parts, so that it can ensure the highly stable measurement of 3D data. And using this hardware that we provide and also software for reconstruction, it takes about 0.1 second for measuring three-dimensional raw data. And by analyzing the reconstruction process, typically it takes about like 5 to 10 seconds, depending on the computing power of your system. And you can measure and retrieve three-dimensional tomograms of the refractive index. As you can see here, it's a single cell surrounded by some high refractive index, the basket, which is like the lipid component. And then you can reconstruct three-dimensional structural information in terms of refractive index. That the meaning of the biological meaning of refractive index is protein concentration, because there is a the linear relationship between refractive index values and protein concentration. So, as I mentioned before, the HCT allows the label-free, three-dimensional, and light-free imaging, then as well as quantitative the analysis of the subcellular organelles. So, not only just providing these three-dimensional images of the cell from those quantitative numbers of refractive index, you can perform the further analysis. Like, for example, you can retrieve the total volume and surface area of cell membrane, and you can also interpret the refractive index as a protein concentration. So you can also calculate the protein concentration as well as dry mass. That can be done using the software in automatic manners, and you can also perform in terms of subcellular organelle levels. So no labeling, because we use refractive index as our image contrast. We don't have to introduce the staining dyes or the fluorescent proteins and etc. Our resolution, spatial resolution, go down to 110 nanometer, because in terms of the three-dimensional reconstruction, some of the high diffraction angles can be considered. So we call as the synthetic aperture effect. So our spatial resolution go beyond the diffraction limited spot size determined by the objective lens numerical apertures. And furthermore, we use the green lasers, and the light transfer or the absorption by the samples is very negligible. So most of light just transmits through the cell, but the energy, there's no energy transfer, but just light is scattered, like the hitting the ball in billiards. So since there's no or the little energy transfer from light to the cell, you can ignore the heat generations or the damages by the illumination source of the HCT. And it takes about like 0.1 to 0.4 seconds for measuring three-dimensional data, followed by the reconstruction process. So you can address very fast dynamics of the cells and tissues. And from those measured three-dimensional refractive index distribution, you can also perform the additional quantification, such as cellular volume, surface area, and total drawing mass, and concentration of proteins can be retrieved in physical, the real numbers. Using this machine, the many researchers have measured very exciting the images and movies of cells and tissues. For example, let me just introduce some representative data. So first one is the primary cultured neuron cell. You can see cell body as well as axon and dendrite. And since you can measure these three-dimensional images without introducing the invasive procedures. You can track the growth of the cells over time in days and up to a few days, as long as our incubators can support the viability of the cells and tissues. Unlike the conventional fluorescent microscope, if you introduce the fluorescent dyes or staining, you have to worry about the cells are not happy and alive anymore. So you have to discard the cell after measuring three-dimensional fluorescent images. And then you have to stain the other samples for the other measurements. Since we can measure multiple 3D data per second, we can access the dynamics of subcellular organelles and the overall cell membrane. And the live cell imaging capability of HT is well suited with the neuroscience as well as the stem cell researchers, because you don't want to modify or induce the unwanted effect to the cell. And subcellular organelles of cancer cell line can be easily visualized and clearly distinguished from the other adjacent the organelles. And for correlative imaging, the experiments like since HCT can provide structural information, and sometimes you want to measure the molecular specific information addressed by the fluorescent microscope. So one of our machine, the model, can measure both 3D, the reflecting index tomograms, as well as the multi-channel, the 3-channel fluorescent 3D data. So you can access both structural and molecular information by measuring the reflective index tomogram as well as the fluorescent tomogram information. So that can also be understood as the PET-CT in medical clinical study imaging. So PET provides molecular information, and CT can provide the structural information. When they use together, it can provide the synergistic effects because they are providing the independent information. And since we can also measure unlabeled the cell in 3D or the time-lapse 3D, you can also see how this target cancer cell is being destroyed, killed by the immunotherapies, in which we have measured the interaction between CAR T cell, chimeric antigen receptor T cell, and the target leukemic B cell. So you can see how they can form the immunotherapy synapse and how this target cell is being destroyed by the process of apoptosis. Due to our high resolution and the three-dimensional label-free imaging capability, we can also provide clear and high content information of the bacterial growth. So what we have measured here is single cell, and the cell has been measured over time, and you can see how they can grow and divide and finally forming the bacterial fields. And using this machine, many researchers have found very interesting application in cell biology, from the cell biology to the hematology, as well as biophysics. And there are a lot of fields can also be addressed by using this holotomography system. Those are the papers from our users last year. And a couple of weeks ago, the group in Princeton has used our machine for the study of the relationship between the phase separation in cell biology and cancer metastasis, published in Cell Biology as a cover articles. Going back to the main topic of this webinar, I'd like to introduce how this HT system can also be utilized for the study of the platelet and their dynamics. As you may know, platelet is one of circulating cells in blood, and which provides the crucial role in the coagulation. So before the activation induced by agonist or the physical stress, the size of or shape of the platelet looks like the red blood cell, very small, but in size in terms of 1 to 2 micrometer in diameter, and they are circulating inside the blood vessel. But once they activated by several the mechanical or chemical or molecular cues, they undergo significant morphological alteration such that it generates like pseudopods, and then it finally forming like a fully squared platelet, so that um, by forming the coagulated forms with other cells and other bio molecules, they can generate the coagulation structures. And finally, the force forming the embolus in the blood vessels. So the right-hand panels, you can see the electron microscope of the individual platelet, and you can see after activation, they are generating the pseudopods, and the morphology is not very smooth in the beginning. The problem in studying the platelet by imaging those shapes is not, there are several challenges. First, the size of platelet is relatively small, and furthermore, the small pseudopods of platelet is very small. Sometimes it is very hard to distinguish it or the recognized by the bright field microscope. And furthermore, the activation process is very fast, from millisecond to second to the minute. So you have to measure the dynamics of movies of this activation process in order to fully address and fully study the mechanism of the activations of platelet. But as I mentioned before, the conventional bright field microscope does not provide the contrast for these platelet as well as other the blood cell. So you can barely see their outline, but in order to enhance the contrast, if you introduce the staining procedure such as H&E or the white Kimsa staining for blood cells, they are not live anymore. So you cannot study the dynamics of the activation process. The resolution limitation also making it hard for study the investigating the small morphological features of platelet. For these reasons, some people also employ electron microscope, even though they can provide high resolution, as you can see in the right-hand side. But again, for the same reason, you cannot address the dynamics of platelet activation. So the size and fast dynamics of the platelet activation is not very suitable to be studied using conventional microscopic technique. That's why several people have introduced the novel imaging techniques such as the TURF or the super resolution and the quantitative 2D quantitative imaging as a layer-free capability in combination with microfluidics, and people also use intraviral microscope for the study of the in vivo process. But those challenges of imaging the platelet can be addressed by the holotomography system. And that's why we wanted to share some of the very early representative study done by our colleagues and the users. So those are the images that we have measured using the holotomography system for the study of the platelet. And in the left panels, you can see the platelet before activation, and after activation in the right-hand panels, you can see the formation of the pores. And for quantitative imaging, the analysis using the ACT system, you can measure the individual platelet and before and after activation, and using the automated analysis software, the Tomocube, you can perform the automatic segmentation from which you can retrieve the volume, surface area. You can retrieve those parameters, including the volume, surface area, project area, the mean refractive index, then protein concentration and dry mass, and etc. And there are several groups that use this machine and the technique for the study of the platelet. Let me first introduce the work done by the groups in University of York, the UK. And they have used the Tomocube system and have measured the three-dimensional shape of individual platelet um, under the genetic the mutations as well as the infection by the parasites. So this is the wide field the imaging of the platelet in 3D. And if you just take out the one of the cell, then you can perform the quantitative imaging analysis. Since we have the three-dimensional tomogram in terms of reflective index, you can retrieve those physical parameters as I mentioned in previous slides. And our group in TIES, we have also studied the activation of platelet and their morphological the alteration during this activation process. And we have first measured the resting platelet, and we have introduced three different agonists, including collagen and thrombin and TRAC. And as you can see, after some time, the cells undergo the significant morphological alteration and finally forming the thin and the spread shape of the platelet. And using different the agonists, you can see how not only the shape of the cell and how the internal structures can also be changed. So mean effective index, or in terms of mean the protein concentration and total dry mass, undergoes a significant decrease by the agonists, including thrombin and TRAP. But as a control system, when you have used collagen, there was the negligible difference between the resting and the collagen condition. Since we can also track the time-lapse 3D in high spatial temporal resolution, we can retrieve the mean effective index as well as other physical parameters in very short period of time duration. So as you look at the figure E, you can see the mean effective index decreased significantly, but some of the representative platelet and indicating that the cells are decreasing its concentration and increasing the volume and forming the spread like the morphological fluctuation. The researchers in University of New York have used the Tomocube HD machine for comparative study between the wild type and the mutations effects of the platelet. So they have measured the three-dimensional effectiveness tomogram of platelet in wild type condition as well as the platelet carrying the mutation called the JAK2V617F. And from those three-dimensional quantitative imaging in terms of reflective index, with several physical parameters, including the dry mass, volume, surface area, projected area, sphericity, and reflective index were retrieved. And they also studied how the platelet shape can be changed if you have the parasite infections. So when they have measured the platelet from the donor infected mice, the platelet demonstrate some of the shape is very very thin shape. So it displays the same diffuse effectiveness distribution, and it becomes very thin, and it also becomes very different in terms of the three-dimensional the shape compared to wild type the platelet. And the related physical parameters can also undergo significant changes compared to the wild type condition. So to summarize, the imaging platelet provides very useful and important information for the treatment of cardiovascular disease and also the assessments of anticoagulation drugs. And previously, people have used a DIC or the other bright field microscope technique, but the limitation is that their limitations are the limited spatial resolution and as well as you cannot access to the dynamics of platelet activation. Higher resolution imaging techniques such as electron microscope cannot access the dynamics of platelet activation as well. But the holotomography system can provide unique advantages in imaging and quantifying individual platelets because it can provide high 3D resolution and without using the exogenous labels or including staining or dyes or the fluorescent proteins. And physical parameters such as the protein density or the protein mass, as well as the dimensional volume and shape, can also be retrieved from those dynamics from this 3D data. And importantly, the dynamics of platelet activation can be fully addressed by using the HT. So this is very interesting and exciting topic of study that can be done by using the HT, but this is very early stage. So a lot of interesting research output should be followed, I expect, including the systematic control of agonists and the responsiveness of individual the platelet to the specific deactivation stimulus simulation would be the direct study. For further information, you may want to refer to the review paper in 3-dimensional and 2-dimensional quantitative phase imaging. And the first part of the paper, the webinar was reproduced from the bioarchive, the preprints from my group. And the major part of this webinar was from this recent paper published in Frontiers in Physiology by the group in the University of York. And you may also refer to recent papers by the Korean University scientists. And they have also used holotomography system for the study of the platelet, the thrombus formation. With that, I'd like to thank for your attention, and I'll be very happy to answer your question. Thank you for your listening.